our next speaker is Stephanie Marcotti, who is going to be talking to us about particle image velocimetry to study cell migration, and she's also going to be doing a demo. So I think we're going to start with some slides, and then we're going to move on to the demo. So I will stop sharing the screen, and I will hand over. You should be able to share your screen now.
Can you see my slides?
Yes.
Great. Yep. So thank you for all for being here. Thank you for the introduction, and thank you to the organizer to allow me to present this work. As Jeremy said, I'm going to give a short intro, about 10 minutes with slides, just to tell you what the software does. And then I'm going to try a live software demo, hoping that everything will work. Keep your fingers crossed. Great. So the first point is, what is particle image velocimetry? Particle image velocimetry, or PIV, as I'm likely to call it for the rest of the talk, it is a tracking method. Instead of tracking single particles, what we do in PIV is we track features within a field of view. It was originally born as an experimental method and an optical method to study fluid flows. In particular, particles were seated inside fluids flowing, and optical tracking of these particles would be used to infer the properties of the fluid. From there, it evolved both computationally and experimentally, and has been used for a number of different applications. What you see here, for example, is the study of birds' flight, and oil droplets are suspended in air where these birds flying to see how the displacement of the air happens around the wings. Today it seems to be like a lot of animals are involved. So after the birds, and the squirrels and the unicorns we've seen before, I'm going to move on to some elephants to explain to you how the software works. So first of all, we need a movie. For my elephants here, I have a really, really short movie. It's just 2 frames, and I'm going back and forth between these 2 frames, and you see our elephants moving in the savannah. So I have an object that's moving. I then have to decide what object I want to track. Since I feel at the moment we all can do with more baby elephants in our life, what I'm going to track is a baby elephant called Cookie that you see in this slide. So for PIV purposes, the first thing I have to do is find Cookie in the first frame. And in this case, I drew a green box around Cookie, which I will call the source box. Next, I have to search for Cookie in the second frame. So I draw another box, which is magenta in this case, which is bigger, and I look around for Cookie by trying to find the point of highest correlation between the 2 images, which I now found, and this is how I found Cookie in the second frame. Once I know the position of Cookie at frame 1 and the position at frame 2, what I can do is to calculate the instance velocity by dividing the displacement between the 2 locations by the time that span between the 2 subsequent frames. So in this version of the software that I'm going to present today, there are plenty out there, all great. I just like mine because it's mine. But in this version of the software, we have to set some parameters. So I'm going to give you an idea on how to choose the parameter to start with in order to start your optimization. The first parameter we have to pick is the source. So the size of the green box that you see in this frame. Going the wrong direction. If it's too small, the software might be confused, as there are many regions of the picture of the frame that have the same characteristics. So for example, here on other elephants or even in the cloud. If it's too big, on the other hand, the software might be as confused because it will not know which one is our object of interest, so which one is Cookie. So a good rule of thumb is to start with the source box, which is proportional to the object we want to track, to the size of the object we want to track. Then we have to move on to the next parameter, which is the search box, which is in magenta in this slide. Remember, this is where we look for our source. If it's too small, we have to make strong assumptions on the direction of motion of the object of interest. So in this case, my assumptions were wrong, and I drew the magenta square behind, so against the direction of motion of the object. So it's unlikely that the code will successfully track cookie. On the other hand, again, if it's too big, there are many elements within the movie that have similar characteristics to cookie, and therefore the code might be confused. Good rule of thumb, source should be half the size of search. So search, double the size of source. I do really apologize for calling these two with two really similar names. I didn't think about it at the time, and then I had to present these and saying these out loud, but now that's how it will be. Third parameter is correlation threshold. So I said we search around the search box for the point of highest correlation for our source. These parameters goes from 0 to 1. 0 being a low value, which means we are not really specific in terms of the object we are tracking, and we are flexible to objects that are similar. On the other hand, if we go high with the correlation threshold, we are really, really specific to the cookie in this case. However, a risk of going too high with the correlation threshold is that if our object of interest has changed slightly, for example, if our elephant is moving its tail, then the software might fail to recognize it. So good rule of thumb, if the values go from 0 to 1, 0.5, it's a good starting point, so somewhere in between. There's another parameter I haven't introduced yet. This is normally the case that we are not tracking just one element in a movie, but we are tracking a number of elements in a movie. So what's the distance between the two elements we want to track is what will define our grid distance. In this particular case, I want to track another baby elephant, which is called Lulu, and I want to know how far is Lulu from Cookie in order to decide how much I should slide the source box to in order to start again the tracking for the next object. Great, enough about elephants, great. Time to move on to cells, and I'm going to introduce another animal. So here you see Drosophila embryo. It's a fruit fly. The embryo is alive and happy, and you see here some cells that we really like that are called macrophages. These cells that you see here, nuclei in magenta and actin cytoskeleton in green. For those of you who are not familiar with cell structure, actin is a network of fibers which is highly involved in the locomotion of cells, and it is really dynamic, as you can see from this movie. We really like these cells because they move in the live embryo roughly in a 2D space, and therefore with microscopy we can obtain movies with really high temporal and spatial resolutions of these cells in vivo. In particular, we recently published a paper in which we characterize the actin flow within these cells, which are moving if I click on them. Here we go. By characterizing the actin flow through PIV and by studying how these correlate to cell motion. Great, so how does the parameters choice translate into real world data? In this case, the actin network is really, really dynamic, especially in these cells that move quite fast. And therefore, we want to use quite small source and search in order to capture the dynamics of this motion. Moreover, we are not just interested in one location, but we are interested in tracking all of the actin flow across the cell. And therefore, we will use a grid distance that's relatively small to cover the whole of the cell. This is an example frame of our PIV analysis in which we're tracking actin. Magenta highlights the outline of the cell, and each green arrow is a vector showing you the instant velocity of that specific actin feature between the two subsequent frames. Real world data are not always as good as ideal world data. Therefore, sometimes we might have some regions that are noisy in terms of signal, or some vectors that are missing because the PIV failed in that specific region. Therefore, an extra step we can perform on this data is some interpolation. Interpolation will allow us to fix these little areas in which the tracking was not performed correctly, and to obtain a complete vector field across the whole object. To interpolate, we need to define two extra parameters, which are the kernel size and the kernel sigma. We use a Gaussian kernel to smooth and interpolate the data, and we can choose the size of this kernel and what weight is given to each region underneath the kernel. If you look at the figures on the right, the colors toward red will give higher weight, and the colors toward blue will give lower weight in a Gaussian size. So these are the final two parameters, and I'm going to show you here the final result of one of our cells. On the left, you see the cell with actin highlighted, moving happily in the fly embryo, and on the right, you can see the tracking of the actin flow during cell migration, and you can see in the color scale the magnitude of the velocity, and the arrows will give you the direction. What I want to highlight, especially for this audience, is that PIV is not limited to one specific cell type in one specific animal, but we can track a lot of different things. I work mainly in biology, so most of the examples are biology related, but we use PIV for different things. For example, to look at actin in different cells, in neuronal growth cones, or even in cell in cultures at high density, or to track tissue compaction, again in the Drosophila embryo, but this is at the tissue scale instead of the cell scale. Great. Now it's time for the software demo. You can find the software at this address on GitHub. Please do get in touch with us in case something doesn't work. That might happen. So here is my email down the bottom left corner, and our Twitter handle should you need to get in touch with us. But now I'm going to try my luck with the software demo. Right, so the first thing is not showing you security and privacy, but going to that GitHub address that I showed you before. So this is my GitHub handle, and this is the PIV repo. One thing I want to mention is there's a pretty good, if I say so myself, README file that includes some information on how you should pre-process your images in order to for the software to run correctly, and some information about the Matlab version you need to use with this specific release. So have a look at these before you start your analysis if you are interested in testing it with your data. I'm pretty sure most of you are familiar with these, but the first thing we need to do is to download the code locally. So I press on this green button called code, and I download the zip file on my desktop. I am going to show you where it is in Finder, and I'm going to unzip it so I can show you what's inside. I have, again, the README file, the license, and a bunch of Matlab functions. All of the code is in Matlab, some of which I'm going to show you how they work in a second. We also need a movie or an input for our tracking. And I'm going to use as a movie one of the cells that I showed you before. So here you can see the cell, and it is a short movie. I just cut it to 10 frames roughly in the interest of time. So again, this is a cell with actin being highlighted, moving in vivo in a Drosophila embryo. Great, time to go into Matlab, navigate to where we saved our code. So in my case, it's in desktop and this PIV master. And the main software in this repo, and the one I'm going to show you today, is this one called Happy PIV. The name is because it will ask the user if they're happy with the test. And at some point I call it Happy to distinguish from other versions, then it sticks, and I kind of like it's called Happy. So Happy PIV, here we go. I will just click run and see what happens. The first thing I'm asked to do is to load the cell movie. So I will press OK, and now I will navigate to the folder where I have my movie and select it. You see that at the moment in the folder, I only have my movie. So I will press open, and I'm asked for an output name. In this case, I'm calling it 0, 1 cell. You can call it however you like it. Just don't put spaces, please. So if I press OK, I will be shown the parameters. These are the parameters that have been optimized for this kind of movie, and the one that I showed you in the slides earlier. However, I'm going to walk through again, and I'm going to show you what happens if I change them. So I have the source and search boxes and the grid distance, which depends on the size of the feature we want to track. Correlation threshold is 0.5. As I said before, go from 0 to 1. To start with, a value in the middle is a good shout. Then we have two parameters, which are the pixel size and the frame interval, which depend on our movie of input. So these will be information that relates to how the movie was how the video was taken in the first place. In this case, we have a pixel size of 0.1 micron and a frame interval of 5 seconds. We have two extra parameters here. One relates to the frame rate to be analyzed. If we leave it to 1, all of the frames will be analyzed, all of the frames in the movies. However, especially in the testing phase, when I optimizing the parameters, I can drop some frames in between. So if this number gets changed to 2, for example, I will analyze frame 1, 3, 5, so every other. Or if I change it to 3, 1, 3, 5, 1, 3, 6. So I will drop 2 frames in between. Why is this helpful? Sometimes the way that the movie was acquired is too fast to see motion. And if I don't see a motion by eye in 2 subsequent frames, then the PIV will not know what to track. So I have to check by eye that 2 subsequent frames show me some motion for the PIV to be able to see it. If this is not the case, because your temporal resolution was too fast when the acquisition was performed, you can run some tests here in the code by dropping subsequent frames to see if the tracking is performed better. You can also pre-process the movie to drop these frames. So both are viable options, but this is sometimes useful, again, in the optimization of the parameters. The last thing will load the total number of frames available to run PIV. In this case, I have 10 frames in the movies. If I have really long movies, especially in the optimization phase, I might want to, let's say I have 100 frames, just want to analyze the first 10, just for ease and in the interest of time again. So I can change this number to a lower number in order to speed up the testing. I'm happy with these parameters, I press OK. I'll be asked if I want to run a test. As I said, these parameters has been already optimized, but I will try and run a test to show you what happens. So I press yes. What happens if I press yes is that the first 2 frames of the movie are being compared. And as you can see here, the vector field is created for the source and the grid distance parameter I selected. What I'm looking for here is that across the cell, I have a homogeneous coverage in terms of green vectors, so that most of the regions of interest within the cell has been tracked correctly. If I have empty patches, it might mean that either our temporal resolution is not great, the signal is not good enough, or overall the PIV is not working correctly. So it might be worth changing your parameters and playing around a bit more. In this case, this doesn't happen, and also the vectors are pointing in the direction I would expect, as I have some underlying notion about the actin flow, and I know it's a retrograde flow from the cell edge, which is this one, to the cell body, which is here. So in this case, everything is working correctly. Thank you very much. Parameters are optimized, but this is what I would look out for. Once I'm happy, I will press OK. And that's why the code is called happy PIV. It will ask me if I'm happy with the correlation test. If I press yes, the analysis will start. But I will press no, just to show you an example of what happens if I change the parameters. So I will increase the size of the source and the size of the search and the grid distance as well. I will leave the correlation threshold as it is. The pixel size and the frame interval depends on the movie, and I will not change the frame rate or the maximum number of frames. Press OK, and I will run a test, yes, to show you what happens. So what happened here is that I have less vectors available. And this is because I increased the size of the source and the search. In this case, the code is still performing correctly, as in I don't have many empty patches. But something to keep in mind is if you increase the size and of the source, you'll also increase the erosion around the edges to avoid edge artifacts. So I'll press OK. I will say I'm not happy this time, just to go back to the optimized parameter, which I'm showing here, and I will press OK. I will not run the test this time, enough. And now the code will start and the analysis will start itself. As you can see in the command window, there's going to be a frame count. We remember we had 10 frames, but it's always a comparison between two subsequent frames. So frame 1 to 2 to 2, 2 to 3, 3 to 4, and so on. So we only have 9 comparison within 10 frames. Press OK to continue, and I'm asked if I want to interpolate. If I'm happy without the interpolation, I will press no, and the code will finish. If I press yes, I will be given the other parameters to set. Kernel size and sigma, I showed you already. I have another option here to set the length of the color scale. So blue is 0, red is whatever number I input here in microns per minute. In this case, I'll leave it to 12. And if you remember from my interpolated movie, what you saw was the color scale, and on top I overlaid some vectors showing the direction of motion of the object to be tracked, so the actin in this case. And I can choose here the spacing between the vectors for visualization. These two are just for visualization purposes. I leave it to 2 and press OK, and a new window will open and the interpolation will happen. Okay, that was quite quick in terms of output. So I'm going to show you what are the outputs that have been created. If you remember here at the beginning of the software demo, I only had my movie. Now 3 folders has been created. One is data, and these contain 2 Matlab files that have the PIV fields for the interpolated and the raw files and the output that I assigned. So I can reuse these for further analysis if I want to. In the images folder, I have the 2 movies that I created and that you quickly saw on screen while I was talking. So if I open them, they are just here. You can see your output and you can check if they are visually appealing and they are showing what you were expecting. Finally, you have a third folder, which is called parameters, which will save all of the parameters that the user inputs while running the code. This is particularly useful if you are going back to some analysis you've done previously, or if you're optimizing your parameters. So you want to know which parameters you use for that specific occurrence of your optimization. So again, this is a Matlab file and contains the output file that you decided, and you can open it in Matlab again and go back to the parameters you've been using. Okay, seems that everything went okay. I am happy to take any questions, and please, if you find yourself maybe looking at the recording of this in the future and have any questions or anything I haven't covered, please do get in touch. Again, you can find us at this email address and Twitter handles, and I am now happy to take any questions. So hands back to Jeremy.
Okay, thank you very much indeed, Stephanie. That was great. So, if people have questions, please do post them in the chat. I'm just going to bring up the slide for this. I think I got the right one. Yes, there we go. So, as I say, please do put questions in the chat. So let me just get the chat window back so I can see. So I had a question about performance and kind of what is the normal. So I mean, I wasn't sure. You showed obviously an example there running over kind of 9 frames. If you were working with a video like this normally, what sort of frame rate would you expect normally, and how many frames would you normally have to analyze to get some kind of useful results?
So in this case, the frame rate is 1 image every 5 seconds. So it's incredibly quick because these cells move fast. But it depends on the cell type. Some cell like 1 every 30 seconds is enough, or 1 every 20 minutes if they don't move quick. And the length of the movie, it really depends what you want to do after in terms of analysis of the vector field. So normally, in the case of the 5 seconds imaging, we would pick movies that were at least 2 minutes in length in order to have an idea on, for example, persistence of motion or averaging the speed, the instant speed across frames. But there's no specific indication on how long or how quick the movie would be. It really depends on the object to be tracked. In terms of performance, the software normally works on 8-bit images, and it's like it is supposed real time, so you saw that it's relatively quick. Of course, if the movie is big, sometimes it happens, for example, for whole tissue movie, the movie itself in terms of dimensions and resolution is really high, and that will affect the timing as well. But it's not normal machine and hopefully normal things that people can access in a lab or with a normal computer.
Okay, thank you. And one other thing that I was curious about, I don't know if I understood correctly, but I think you said that the movement has to be kind of fairly consistent in terms of between frames. So if you had, can you have a case where effectively you need to change your parameters between frames, and how do you handle that? So can you have a case where something moves much faster in one case than in one set of frames than another? Do you end up with search space that's too small or too large or whatever? And how do you work around those kind of?
That's a very good question, and something I've never seen despite trying to run PIV on almost everything that passes through the lab. So I never had the case of different motions across the movie, but I can't see why that couldn't happen. The code is not optimized to change a parameter halfway through, so you would need to divide the movie into 2 parts and run 2 separate sets of so the PIV twice on the 2 separate chunks of the movie. The only problem with that would be that then if you wanted to compare the two, you would have to be really mindful in terms of not comparing apples with potatoes and making sure that, for example, the temporal resolution is the same, or that if the window size is different, you use global measurements and not local measurements in order to, sorry, for example, flow across the old field of view in order to compare the 2 chunks of the movies. But I've never tested because I've never had such example.
Okay. I don't know if we have any other questions. I have another question, but I don't want to monopolize the questions other people have got. If other people have got questions, so if anybody else has got anything, please do put it in the chat, or if you want to unmute and ask a question, you're very welcome to. So while we're waiting, I will just ask my last question, which is I'm curious to know whether this is an area where you know if people are doing anything with machine learning, because it sounds like this kind of problem potentially sounds like something that might be tackled through machine learning ideas. And also, if you get things where you need to change your parameters, is that potentially something I guess you could learn with that? Is that something that you've looked at all, or do you know if that's an area that people are looking at?
It's definitely a new thing for biology. A few groups are starting to work in that. There's a really strong group in Germany with Florian Jug that's working into that a lot, and they are building some incredible pieces of software, especially Fiji macros, in order to, for example, segment automatically using machine learning. And run some denoising of imaging to increase the resolution of microscopy images that are not as good as they could be, but it's relatively new. Consider that most of these analysis that I showed you has been done manually before a couple of years ago. So this is already something that is quite advanced for biology at the moment. It's definitely changing, and it's changing really fast. So I'm sure that machine learning will overtake my PIV really quickly. But for now, I haven't personally worked on these, on machine learning applied to biology. But that's definitely the way forward, I'm sure.
Okay, thank you very much.